it as a project under the 16-week Open Science Mentorship, Open Life Science Organization. So over the course of 19 weeks, the initial ideas were further developed through the OLS mentorship program. Also, through this, we have managed to reach out to a much broader audience and raise visibility on the initiative. Finally, all of this wouldn't happen without your support and contribution. We would like to thank our amazing speaker today, Dr. Natalie, and moderator, Dr. Hari, for accepting our invitation, Prof. Tinvey for taking time to give an inaugural speech today and attend the talk, APBioNet President, Dr. Asif, for his inspiring leadership and endless support, APBioNet contributors, Ms. Debangana, Ms. Chong, Ms. Rizkiana, Dr. Diksha, Dr. Sharanya, for their amazing hard work and teamwork. Also, OLS community and leaders, OLS mentors, Ms. Patricia, and everyone. Thank you very much and enjoy the talk.
Thank you, Ms. Helia. Next, we invite the founding convener of APBioNet, Professor Tinvey.
Hello, everyone. My name is Tinvey. When I was a young man in 1998, I got together a few of my colleagues in order to set up the Asia Pacific Bioinformatics Network. And 22 years later, lo and behold, I'm surrounded by so many young people. I'm really delighted to see so many young people stepping forward in order to promote bioinformatics in this community. 22 years ago, there was a dire need to introduce bioinformatics amongst life science researchers. Today, we can see that it's a thriving environment and that there are so many speakers from all over the world wanting to help us within the Asian Pacific region in order to promote bioinformatics amongst yet another new generation of young people. So I'm really honored and delighted that we are able to have this event and to continue and to champion bioinformatics throughout our region. We are indeed honored to have Professor Natalie to be able to give us this keynote speech, and Professor Hari as well. And I'm really happy to see that my ex-PhD student is now again voted and elected president of APBioNet yet again. And we are really happy to see that generation after generation of students have now become professionals and our peers, and hopefully to be able to succeed in their careers in this line going forward into the future. So on this regard, may I wish all of you a very successful APBioNet talk, and we're really happy to see all of you come together to put together such a wonderful webinar series. So on that note, have a great time, have a great learning experience. Thank you so much, all of you, for working together to make bioinformatics a thriving discipline in our region. Thank you.
Thank you, Professor Tinvey. We now move on to the talk by Dr. Natalie Twine. Before we begin, I will invite Dr. Ranji Hari Nair as our moderator. Dr. Hari, the screen is yours.
Thank you, Chakraborty. And I'd like to welcome everyone for this APBioNet inaugural talk by Dr. Natalie Twine. So before we move on further for the talk, I'd like to introduce our speaker, Dr. Natalie. Dr. Natalie Twine is the team lead of the Genome Insights team within CSIRO, that is the Commonwealth Scientific and Industrial Research Organization that has many branches in Australia. She focuses on developing technology for population scale genomics. Dr. Twine's research career to date has focused on translational and clinical genomics, and she's passionate about using the technology to impact health and understanding complex diseases. And Dr. Twine thrives on collaborative research and mostly collaborates with Macquarie University and the international project MinE Consortium, and others. Her publication record is strong with 26 peer-reviewed publications, over 1,030 citations so far, and she has a H index of 14. Dr. Twine has presented at numerous international conferences, including ISMB 2019 Switzerland and MND Symposium 2019 Perth. She obtained her PhD in bioinformatics from University of New South Wales and has previously worked for UNSW, King's College London, and University College London. So with a wide array of experience, I believe that all of us will learn a lot from her talk today. So without further ado, I'd like to invite her for delivering her talk today. Welcome. With a warm welcome, we'd like to welcome you, Dr. Natalie. The stage is yours.
Thank you, Ranji. And welcome everyone. Thanks for joining in. And I know we're getting close to the holidays now, so I'm really appreciative that everyone's taken the time to register and log in and listen to what I have to say today. So I am going to start by sharing my talk. All right, can everyone see my screen? Okay, so I'm going to talk to you today about revolutionizing genome analysis using machine learning and cloud computing. So as Ranji said, and with his very thoughtful introduction, I am from the CSIRO, or the Commonwealth Scientific and Industrial Research Organization. So we are based in Australia. We are Australia's largest national research agency, and well known for our invention of Wi-Fi, which is used globally, also for the development of the Hendra virus vaccine, as well as a number of very popular diets used across Australia, including the total well-being diet. And within CSIRO, I work within the Australian EHealth Research Centre, and our vision is to really improve health and healthcare through digital technology and services. So my talk today is focusing on genomics and technology we've developed around looking at genomic data. So genomics affects everything in our daily lives, from how we look, to our disease risk, and even to our behavior. For example, whether or not we have a hitchhiker's thumb. So a hitchhiker's thumb is the ability to bend your thumb all the way back. I don't have a hitchhiker's thumb, but 1 in 6 of us do. And this is by virtue of a genetic mutation that you may or may not carry that enables you to have this particular physical phenotype. So genomics can also affect our disease risk, so whether or not we have healthy lungs, or in fact lungs affected by cystic fibrosis. This is by virtue of genetic mutation. And then also by our behavior. So in fact, whether or not we like the taste of coriander. So many people dislike the taste of coriander. Around 1/3 of us, in fact, dislike the taste of coriander. And this is again due to genetic mutation. So genomic data is growing exponentially, and it is vast. So if we were to sort of measure out the size of the DNA in our cells, so we have around 2 meters of DNA per genome. If we were to stretch out the DNA, unravel the DNA and stretch it out, and then multiply this by how many cells we have in our body, so around 100 trillion cells in our body, we can quickly see that the information sort of contained within us is in fact larger than the information contained within the universe. So we might even say that genomic data sizes are becoming genomically large rather than astronomically large. So within my team and within our group at CSIRO, we're cloud native innovators. So we really like to make the most of cloud technology when building our platforms. And I'm going to tell you a little bit about the platforms that we've developed over the last few years today. firstly, I'll talk about VariantSpark, which is a machine learning approach with genomic variant analysis. And this helps us to identify disease genes in billions of DNA molecules. Secondly, I'm going to talk about Tribes, which is a platform for discovering genetic ancestry. So here we can discover distant relatives through genome sequences, and again, use this to help us uncover disease genes. And then finally, I'm going to talk about a little bit about our response towards the COVID-19 pandemic and what we've learned from the COVID-19 pandemic, and in fact, what technology we've developed to help in the fight against the pandemic. So as I said, we really like to leverage cloud technology. And I'm sure everyone here has heard of the cloud. And a little example, or a little sort of analogy that I like to use to explain what cloud is and how cloud works is, we liken the cloud to an in-room sound system. And this is because if we look at, if we bring along, so rather than buying our own sound system, we can in fact just rent the sound system. So we bring along our iPad with our music that we want to play, and we plug it into an existing sound system. So we don't in fact need to own the surround sound system. This is much akin to renting compute infrastructure when we need it. So we come in and plug in our software to existing infrastructure that we just need to use on demand. And again, if we were to require more infrastructure, so more powerful infrastructure, so powerful speakers and some woofers, we can again rent these to boost our infrastructure. And much like in the cloud computing environment, we can boost the number of CPUs we have or add GPUs to really boost the power that we have and the grunt work that we have for running and the software that we have of interest. So one particular application case that we have been working on within our group is looking at ALS, or motor neuron disease. So we've been performing groundbreaking research using our latest technology in collaboration with the world's largest ALS consortium called Project Mine. So ALS is a devastating motor neuron disease which leads to death within 2 years and has no known treatment. And together with Project Mine, we have access to up to 25,000 patient genomes, so case and control genomes from ALS patients. So if we use this toy example for looking at disease genes, we can really help to illustrate the problem that we're facing and sort of the technology that we're developing, what the problem is that we're trying to address with the technology that we're developing. So if we look on the left-hand side and we imagine every individual's genome is represented by a blue line, and the genes in the genome are represented by the boxes or the rectangles colored along the blue line. Then within each gene, within genes, we have DNA variants or genetic variants. And what we're really looking for is differences between individuals who are sick and individuals who are healthy as a group to identify which genetic variants are associated with those individuals that are sick compared to those which are healthy. And in this toy example, it's quite clear to see which genetic variant is present in the sick individuals but absent from the healthy individuals. But the reality is when we're looking at complex diseases such as ALS, or in fact, many of the diseases that are prevalent today, such as heart disease, diabetes, rheumatoid arthritis, cancer, these diseases are driven by multiple genes, and multiple genes that in fact interact with each other in a variable way. So really to try and identify these multiple variants and also how they interact with each other, we need a more sophisticated approach to genetic variant analysis. And we use a machine learning approach such as the random forests algorithm for in machine learning. And by using this approach, our research question at hand is to look at we can look at 1.7 trillion data points. So we have, as I said, this cohort of individual genomes from the Project Mine consortium, where we have around 22,000 individuals' genomes. And across each genome, we have 80 million features. And then we have the disease label, so either whether or not the individuals are sick or healthy. And we developed a solution which can really cater for analyzing such large-scale data, so these 1.7 trillion data points. And it's worth pointing out here that the technology that we developed doesn't specifically have to focus on genomic data. In fact, we can look at any type of data that fits this format, such as time series data, sensor data, or concatenated data. And also the outcome doesn't necessarily need to be disease status or healthy or disease status. It could also be looking at a business's churn rate, the occurrence of failure, or indeed the product quality. And we will then want to identify what are the predictive markers, in this case, the predictive genes that are driving a particular outcome, or in the case of a business solution, what are the predictive factors that are really driving a particular outcome. So when looking at population scale data or population scale genomic data, we really need a big data solution. So looking at the trillions of data points, we can't use sort of standard computer infrastructure to really analyze this scale of data. So the majority of bioinformatics applications will use a sort of high performance compute cluster approach, which really has a compute intensive focus. So it is bound by a specific number of nodes. It is parallelizable to some extent. But the approach that we're really leveraging in our technology is the APBioNET technology. So this has a data-intensive approach rather than a compute-intensive approach. So it's really designed for looking at big data sets. And it's not bound by a specific number of nodes. So it's highly parallelizable and scalable to large data sets. So the technology we've developed is called VariantSpark. So this is a machine learning approach for genomic variant analysis. It has the capability of looking at 3,000 individuals with 80 million features in just 30 minutes. So it's faster than existing tools. It will also have shown in benchmarking, which you can see on the top left, that we outperform other tools, other random forest-based machine learning tools in terms of accuracy as well as speed, when we compare them to other sort of random forest-based machine learning platforms for genomic variant analysis. So one of the aspects that we do, as I mentioned earlier, we like to use cloud technologies to really make our technology available for everyone to use. And it also means that groups or individuals don't need to have their own compute infrastructure set up to be able to use these tools. So VariantSpark is available on all major cloud providers, including Amazon, Microsoft Azure. And it's accessible also through GitHub. So individuals can install it themselves if they choose to through GitHub, but it's freely available there. So recently, we've also put a VariantSpark on the Amazon Marketplace. So we're in fact the first public sector organization to use a genomics product to install a genomics product on Amazon AWS Marketplace. And this work was publicized in our local media, the Financial Review in Australia, if any of you are familiar with it. And this really enables individuals around the world to have access to this, to the tool. And it democratizes technology. So it enables anyone who would like to use the technology, doesn't have to have a computer infrastructure. They can log in to the marketplace, use the tool for the time period that suits them, download their results. And this makes it really a great user experience. So what we've done to really demonstrate our technology is we've built some demo notebooks on Databricks. So if you head over to the URL on the screen, on Databricks, we've set up an instance of VariantSpark, which focuses on identifying the hipsteriness of your genome. So we've been able to calculate a genetic hipster index based on an individual's genome. So in this notebook, we have public genomic data available. So from 1,000 genomes, you can look at any genome from 1,000 genomes, or you can indeed upload your own genome or any genomic data that you have available. And what VariantSpark does in this demo notebook is look for 4 key variants which are predictive of an individual being a hipster or not. So obviously this is a fake phenotype, this is a synthetic phenotype just to illustrate the capability of our technology, and we thought it was quite a fun example just to demonstrate how the tool works and the type of outcome that we get. So here we're focusing on 4 key variants, which are a propensity for identifying check shirts, a propensity for metabolizing caffeine, and also a propensity for hairiness. So yeah, I encourage you all to go over to Databricks and check that out and have a play with that yourself. It was quite a fun example. So moving from VariantSpark and population scale genomic analysis to Ancestry. So we know that disease is often inherited. For example, the risk of breast cancer or heart disease and diabetes are all partially inherited. However, inheritance patterns are quite complex, and really to understand how diseases are passed from one generation to each other, we really need to delve into the inheritance between generations. So here I'm going to talk about our platform, the second platform today called Tribes, which is an ancestry-based platform for identifying distant relatives based on the DNA to uncover disease genes. So by 2025, we estimate that around half the world's population will have been sequenced. And this means that large data sets from genomics will by far dwarf the currently other main sources of big data, such as YouTube, astronomy, and Twitter. So these are currently the biggest source of data, big data. So by 2025, genomics will completely dwarf these main sources of big data. And such large data sets are really going to have the availability of such large amounts of genomic data are really going to have wide-reaching impacts. And one of these impacts is the ability to discover genetic ancestry between individuals. So just an interesting news story. So using genetic ancestry, police were able to identify a serial killer that had been on the loose for 20-odd years over in California, the Golden State serial killer, using genetic ancestry of a distant relative of the serial killer. And an unpublic database called GEDmatch. So by genetic ancestry, we mean that we identify unknown human relationships based on underlying genomic sequence data. So moving from serial killers to more noble causes, we have been using genetic ancestry, again in the application case of looking at finding a cure for motor neuron disease or amyotrophic lateral sclerosis. So this disease was popularized by the Ice Bucket Challenge a number of years ago in social media. You may all remember it, when everyone was challenged to chuck a bucket of ice over their head to raise funds for the disease research. As I said earlier, it's a devastating motor neuron disease which leads to death within 2 years. And a project we've been involved in the last few years is looking at genomic data from around 850 Australians suffering from ALS. So a key concept for looking at genetic ancestry is that of identity by descent. So what we want to do in Ancestry is really identify the segments of DNA that are shared between 2 or more people. So we all share segments of DNA between us, even by the fact that we're from the same population group. But what's key in looking for identity by descent or genetic ancestry is really that those segments that are shared between individuals need to be inherited by a recent common ancestor. So if we look at the example on the left-hand side, we can see the orange segment that's highlighted between the gray lines. These individuals share the segment by identity by descent. And they share them by virtue of their recent common ancestor, which is their grandparent here. So if we imagine, so these colored bars represent chromosomes in an individual's genome. We can share that a segment of this chromosome has been inherited by offspring of this individual and then passed down to the next generation, so the grandchildren in this instance. And so we can tell that these individuals are related to each other due to their shared segment of DNA. So you might ask, well, why are we interested in looking at genetic ancestry? Why is this interesting to us other than to look for our distant relatives? Well, knowing shared segments that are knowing the segments that are shared between individuals, specifically individuals who are affected by a disease, this can really help us to narrow down the search area in the disease region. So instead of just looking for genetic variants, like I pointed out earlier, that are shared between shared within sick individuals, but not within healthy individuals, we can further narrow down the search area by looking for these shared regions between individuals that are related to each other. So these shared segments help us to narrow the search area for disease genes. So the technology we've developed is called Tribes. Tribes is a population scale ancestry predictor tool. It's a population scale tool for discovering relatedness. It's a pipeline-based tool, and it uses a phase-free approach, which means that the genomic data doesn't need to be phased prior to ancestry prediction. And this means that it's really quick to analyze. And it's also, phasing is usually a time-limiting step in the analysis. It also means that it's highly scalable in that we can analyze any amount of shared pairwise relationships between large cohorts of individuals, so population scale individuals. We've also shown the Tribes is more accurate than gold standard tools, and you can see on the plot on the right-hand side, sort of gold standard tool called King, which is widely used for looking at close relationships in genomic data, usually up to third degree. We can see here that Tribes outperforms King at a number of degrees, from first degree, which is a sort of parent-offspring relationship, up to a 6th and 7th degree, which is a more distant cousin relationship. And as I said earlier, by identifying these relationships, we can really increase our power to discover disease genes. So Tribes is available on Docker, it's available on GitHub, and we've more recently also uploaded Tribes. Tribes is available on AWS as a cloud platform, so individuals don't need to install the tool themselves. But if anyone is interested in trying it, please get in touch, because it is available on through a number of avenues. So using Tribes on our Australian ALS cohort, we published work earlier this year where we were able to identify founder events in the Australian population and link individuals carrying ALS to each other. So we'd be able to link a number of families, all known to have individuals, so completely unrelated families. Using Tribes, we're able to connect these families to each other, as well as individuals who apparently had sporadic ALS. So they had no known family history of disease. We were showed that they were in fact distantly related to other families with a known family history of the disease. So what you can see on our analysis on the left-hand side, we've performed clustering of the individuals based on whether or not they're identical by descent over a known ALS gene called the SOD1 gene. So every individual is represented by a colored shape, so these colored squares here, and their color represents the family of origin. So you can see there's multiple families that have been reported to the clinic. Some of these families are small and some of the families are large. And by using our Tribes analysis, so identifying the related segments between individuals, we'll be able to identify which of these individuals are related to each other at this particular locus, so where they share segments of DNA across the SOD1 gene. And you can see here that we'll be able to connect 19 distinct families as all having a common ancestor, having shared ancestry. And they also all share the same genetic variant in the SOD1 gene. So this is quite an important finding to us. You can also see in the rest of the clustering figure that we'll be able to cluster a number of other individuals in the distinct cluster. So in the top cluster, we have individuals which are identified by circles. The gray edges identify that they all share a segment over this SOD1 gene. And what's clear here is that these individuals have a different mutation in the SOD1 gene compared to the cluster on the bottom left, which means that they have a different common ancestor to each other from the cluster at the bottom left. Again, with the remaining three clusters, we show the same thing, that the individuals have the common ancestor And there are in fact five distinct common ancestors for these individuals, these hundreds of individuals with this SOD1 mutation in the Australian population. And you might ask, well, why do we care that there's 5 distinct mutations? So 5 distinct founder effects leading to different mutations in the ALS patients. Well, it's important to know different mutations that are present in a particular gene, because individuals can respond differently to the same drug depending on what variant or what genetic mutation they have of a particular gene. So using tribes, we'll be able to uncover and identify 53 new relatives up to 7th degree in our 800 Australian patients. And by using the shared DNA segments, we can really help us to reduce the search space for candidate disease variants. So using this approach, we were able to narrow down our search space to just 0.2% of the genome. And in doing so, we're able to identify 2 novel disease genes, which we were sent for further investigation in ALS, FIG4, and DYNC1H1. So more recent work that we've been doing in the last 6 months with tribes is to scale tribes up to population scale data sets, to test tribes on large scale data sets to see how well it performs. And again, we've used this Project Mind cohort where we have performed pairwise relatedness analysis across 9,000 individuals, 9,000 genomes. And when we cluster the relatedness results between these individuals, we can see that the clustering shows clear population groupings. So on the left-hand side, again, every shape, every circle represents an individual. The lines between the circle represent the distance of the relationship between an individual to each other. So short lines indicate a close relationship, long lines indicate a more distant relationship. And then we colored them by population origin. So in this consortium, we have 13 countries of origin. And I've shown the major countries here in this figure on the left-hand side. So the Dutch or the Netherlands are colored in pink, Belgium are colored in black, and we can see those population groups clustering close together, which is not surprising given their geographic proximity. On the top right, we have the British colored in green, the Irish colored in orange, and the US individuals colored in purple. And again, we can see them clustering together, which is also not surprising. What's interesting here is that the Irish seem to be more homozygous compared to the British population. On the bottom left, we have the Israeli cluster, individuals colored in red are Israeli of origin. And on the bottom right, we have a cluster of individuals which are Swedish in origin. And what was really interesting here is that the Swedes are very distinct from other individuals, other European individuals. And in fact, they are very homogeneous in nature, showing a lot more close relationships than we're seeing in other population groups. So this is very new work we've been doing on the Project Mind data, because they've just released the latest data freeze. And what we're doing now is looking for shared IBD within these novel relationships to really help us look for new disease regions or new disease variants in this Project Mind data set. So moving on to my last section of the talk today, so some other work we've been doing in 2020 is that around how to address the COVID-19 pandemic. So technology we've developed in response to the COVID-19 pandemic. So we've developed a number of different technologies around monitoring and tracking of and as well as diagnostics for the COVID-19 virus. So today what I'm going to talk to you about is the work we've been doing around monitoring and around tracking of the virus. So COVID-19, as you are probably all aware, is caused by the SARS-CoV-2 virus. It's a 30,000 letter RNA virus, and it's an obligatory parasite, which means that it needs to infect a host cell to be able to survive and to replicate and infect another individual. So all RNA viruses mutate, and this is common to all viruses. And what's key is that the mutation rate in coronavirus is in fact quite slower than for example, other viruses such as influenza. So if we liken mutation rates to copying, so mutation rates occur when a virus is copied. So if we liken the copying of a coronavirus to using a cookie cutter approach to copy the virus, so the copying process is quite efficient, meaning that each copy of the virus is quite similar to its parent, the parent virus. So the mutation, the copying includes proofreading, which means that the copies are quite similar to each other. And we compare this to sort of the copying approach that an influenza virus might use, which uses a knife. And you can see that clearly that the copies of the virus are quite different from the parent virus because they just have a higher mutation rate in this copying process. So work we've been doing is looking at the genetic fingerprint of the virus. So looking at so the genetic variants that occur in the virus by taking the genetic fingerprint of the virus and then creating a spatial map of these different strains of the virus. So if you imagine 3 different strains of the virus and compare, what we've done is compare their genetic fingerprint to each other by comparing how similar or different the viruses are to each other based on their genetic variants. And then what we've done is use a machine learning approach to compare the genetic fingerprints of each other in a 3D evolutionary space. So this is the methodology we've applied to 181 strains of the SARS-CoV-2 virus and compared the genetic fingerprints to each other in this evolutionary landscape. And using this approach, we were able to identify 6 strains which we could recommend for vaccine testing and vaccine development in Australia. And these are representative strains that we recommended to our collaborators down in Geelong, which is a vaccine facility in Australia, to use in the development of the vaccine. And we published this work earlier this year in Transboundary and Emerging Diseases. So we've also developed a web service which incorporates this approach that I've talked about, where we look at the genetic signature of the viruses causing vaccines. And we've partnered with GISAID, which is the international database, the international organization for collecting coronavirus strains. So the web service continually monitors new coronavirus strains, which are updated daily by GISAID. And it offers continuous reanalysis of the genetic strains and the genetic fingerprints that exist in the database based on the nightly deploys that GISAID performs with new strains that are submitted to the database. So we've used cloud technology for building this web service, for building our COVID-19 web service. So the cloud technology uses a serverless approach, and a serverless I'll explain to you a little bit in more detail in the next slide. But the serverless basically uses a Lambda function which monitors updates from GISAID. And when there's update from the GISAID database, it pulls this information in. And this then triggers an EC2 instance, which is a new instance of a compute environment, analysis environment, which then performs a comparative analysis of these new viral strains with the existing viral strains that have already been pulled from the database on previous days. This then triggers, this comparative analysis is then output to our website. So as I described earlier, I likened our cloud computing environment to sort of a home sound system where you can plug and play your software of interest, add sort of power to the sound system as needed. And if we take this one step further and you look at serverless, so cloud is flexible, as we know, there's no overhead, it's very scalable. So it's sort of akin to having a chauffeur instead of having your own car. But taking this one step further to the service technology, it really enables us to have agile technology. So it's really cost effective compared to using standard cloud technology. So if we compare, again, using the car and the chauffeur analogy, this would be akin to using a ride sharing app such as Uber. So it's really cost effective. We're only using the compute when we need it. It's triggered, as I said, it's triggered by the daily updates by GIZ, and we're really only paying for the technology when it's needed. So this really, it keeps the cost down and makes the technology highly agile, and it's really suitable for these workloads which are not consistent, but they're sort of bursty in nature. So some other work that we've been doing is monitoring the spread of the virus, and specifically across Australia, this has helped to support the health response. So within communities, it's important to monitor any new viruses that are any new mutations that are developing within the viral strains. So this is similar to something like molecular tracking or even contact tracing in the population, in the community. So we're interested in looking at how the viruses spread across the community and what these mutations are. So we're interested in the proportion of these mutations in the population, and specific global distributions or local distributions of these new mutations. So the technology we've developed around this is called the COVID-19 Beacon tool. This is a project for tracking mutations. It's a joint project with India, with the IGIB Institute in India. And here we continually have built a web tool, again using serverless technology, which continually monitors new mutations that arise in the data sets, the underlying data sets. So looking at tracking mutations is important because new mutations can affect the phenotype of the virus and the way that the virus affects an individual. For example, the D614 mutation, which is now widely established as a worldwide dominant mutation, is well known to make the virus more transmissible. And this arose and evolved to be a dominant mutation quite quickly across the population. And these mutations new mutations are occurring all the time. And it's really important to monitor new mutations that are coming out and their frequency within cohorts and within countries and within regions within countries. So in order to monitor this quickly, we really need fast queries that are possible to establish variant frequencies across distributed files across different centers around the country. So these are totally distributed data sets. So we've achieved this with our COVID-19 Beacon platform, where an individual can query, use the query function to search for a particular viral mutation, look at the frequency in any in subpopulations around the country of interest. So in this project, we've partnered with India, as I said. We're also extending this work to look at virus mutations in China and other parts of Asia. So again, COVID-19 Beacon tool uses serverless technology where we have an API gateway which interacts with the user. This then triggers, again, a Lambda function which sets up an EC2 instance to search for the new mutations. And then that's returned to the web page for the individual to view. So 3 things I'd like you to remember from my talk today. And that is that datafication, or this explosion of genomic data that's occurring around us and is going to continue to occur for the near future, really demands not just big data analysis, but wide machine learning solutions. And technology we've developed in this for this demand is called VariantSpark. This is a cloud-based tool as well as available on GitHub, and it's really available for people to use. Also, with the explosion of genomic data, the ability to uncover ancestry and genetic ancestry is increasing. And by uncovering distant ancestry, we can really help to uncover disease genes that are underlying driving diseases. So the technology we developed around this is called Tribes, and this is also publicly available and is more recently available with AWS, so it's a cloud-based platform. And then finally, the work I've just talked to you today about is that there's a lot of technology in the IT space that can really make a difference in the fight against COVID-19. And I think for future pandemics, you probably agree with me that for future pandemics, really leveraging technology can make a big difference in the response and the outcome of the pandemic to the world. And that's it for me. So I'd just like to thank the rest of my group, and please get in touch if there's anything interesting that anything you're interested in today that you'd like to discuss further, or indeed that you'd like to know more about, or you'd like to work together with us. So we're always collaborating and looking for people who are interested in our technology and who have data sets that they'd like to explore with our technology. So you can go and check out our website. There's a QR code that'll take you there. And yeah, get in touch. And any questions? Thank you.
Thank you, Dr. Natalie. It was a wonderful, enlightening talk. I hope all of us got a lot of learnings from that talk. I certainly did. There are a few questions already from the chat group. Please also feel free to voice out your own questions. Yeah. So what I'll do right now is I'll take the first chat group question, and along the way, if you'd like to ask a question, please raise a hand and I'll give you a go. So I'll start with a more general question from Manisha Pritam, right? She asks us that why are we using machine learning approaches, and how sure you are that these predictions are accurate, since she's pretty new to this machine learning area. So could you give her some pointers?
Sure. Well, I think the major advantage for us for using machine learning, at least in the genomic space, to looking for drivers of disease, is that really with sort of traditional approaches, so traditional statistic approaches which is what the genetics community has used over the last 10 or 20 years, is that it limits the ability to look for variants that interact with each other. So variants, as we know sort of common diseases like heart disease, diabetes, are driven by not just one genetic variant, they're driven by multiple variants. And these variants don't independently contribute to the disease. So they're interacting with each other to drive the outcome. And using classical approaches with statistics, it's really not possible to identify these interactions between variants, because the assumption that these classical approaches make is that every variant is independent of each other, and that's how the test is performed. But using machine learning, or indeed this random forest approach that we have used, which is sort of quite a well-established machine learning algorithm, it's been around for quite a while, you can really the random forest can really discover sort of variants that are together driving the phenotype, so how they interact with each other to drive the outcome. So it's adding additional layers of information from sort of more classical statistic approaches for genetic variant analysis. And I think the second part of the question, can you remind me what that was? It was machine learning and
on the accuracy, how sure you are that whatever that's coming out is accurate.
Yes. So I guess yes, that's a very good question. And with any genetic analysis, I mean, it doesn't matter what your approach is, whether it's machine learning or sort of parametric statistics, validation is important. So replication of the analysis is important, and that we use the same approach that any other methodology would use, in that we have to our results need to be replicable, and they are. So that means we use an independent data set which have a completely different group of samples with the same disease to see whether or not we get the same result. And that's what we find indeed. So yes, we perform validation studies in independent data sets to show that our results are accurate.
Okay, thank you, Dr. Natalie. We do have other questions, but I'd like to ask the group, do you want to ask in person? Anyone? You can unmute yourself and go ahead.
Hi, this is Paul. Can you hear me?
Yes, Paul,
I
go
can,
ahead.
Yeah,
yes.
hi. I am quite drawn to your capability of using VariantSpark. Uh, I know that there are about 80 million features that run over 3,000 individuals in 30 minutes. Is that correct?
Yes.
So, okay. So how much does it cost to run? And how many nodes that you use for that particular analysis?
That's a good question. So the cost is sort of within the dollars. It's within the dollars amount, within the dollar amounts. I mean, in Australian dollars, it's under $10. So it's very cost effective because because you're only really running the analysis for a reasonably short amount of time. So it's just the 30 minutes. For the number of nodes used, I can't remember off the top of my head, but I think if you get in touch offline, we can chat about this offline. I can get the details back to you about that, because it also depends on the depth of analysis that you want to use. So there are some parameters that are sort of tweakable within the VariantSpark analysis. So for example, how many trees you build in the random forest and the depth of the trees that you're going to. And there's sort of a sweet spot you can figure out with that in terms of trade-off of accuracy versus cost and time. So I guess the answer to that is there's not an exact answer to that. It depends on the trade-offs that you're willing to make. But I'm happy to discuss this offline with you if you'd like to test the software yourself or find out more about it.
Yeah, thank you. I think I have a few cases that people actually run in the 10 of thousands. So, so I think can
What
be
type
very
of data
expensive.
sets are you looking at?
That's that. I think to give it to the group. But never mind. I think we'll catch up with you on your runs. Thank you.
Yeah, sure. Please get in touch. My email. I'll put my email in the chat group. I'll send it to you on the chat.
Thank you.
No worries.
So I personally have a related question to VariantSpark. I was like really wondering, what is the 80 million features? That's a lot of features.
That's
so
right. I've lost you. Are you still
of the
there?
features, is it distance
Ranji?
across?
Sorry. You might need to repeat your question because you cut out.
Okay, could you give us some examples of the features? How did you expand it from the genome? How do you encode
sure.
those features? It's part of feature engineering, I suppose, but I'm really curious.
That's right. So the 80 million features we're referring to here are the variants, so the genetic variants. As you know, within the genome, there's positions in the genome that are variable. So these SNPs, single nucleotide polymorphisms. And so what we focus on is those regions of the genome. So this genetic variant, the variable regions, because a lot of the genome is not particularly variable between individuals. So in our cohort of so across these 25,000 individuals in the Project Wine data set, there's around 80 million positions within the genome that are variable enough across the groups, across the cohort, that it's useful for, they provide useful information in terms of features. So that's how we've selected them basically. Within our Australian sub-cohort. So we had, as I said in my presentation, around 800 individuals. So around there, we see, say, 42 million variants. So the variant number will change depending on your size of your cohort, as well as how deeply the data has been sequenced. So if you're looking at array-based data, for example, you see much fewer variants because of the technology that's used. With the whole genome sequencing data, you'll have ability to look at the millions of variants when you're looking at population scales in the order of 20,000 individuals. And then in terms of encoding the variants, it's quite straightforward. So we've just encoded variants which are biallelic, so they can only be one of three. So homozygous wild type, a heterozygote, or a homozygous alternate. So there's only basically, you encode them using 0 and 1, so the individual has to be 1, 1, 1, 0, or 0, 0. And then using a matrix built on that information, we run the random forests application. So the encoding is quite straightforward.
So, all right, purely on the positional of information on the variants. So you don't consider the relative positions
That's
of
right.
those variants. Okay.
Yeah. So, well, I mean, every variant has an RSID, as you know, so it has a unique ID, a unique identifier for every variant. We're not including, so there's no covariates included in the model, such as, I know with sort of other genome-wide associatedness studies, you can look at sort of population background, you can include as a covariate, or like gender, you can include as covariance, but we don't include any other covariate information. So every variant is, there's no information as to the geographical position or the location position of them on the chromosome.
Okay.
But that can be analyzed sort of as a post-analysis. So we can look at which variants are coming up as top hits and seeing, we then know exactly where those variants are in the genome.
Another related question, I managed to quickly look up on VariantSpark, and I think this part where you didn't cover. You're able to somehow, once you get a prediction, you'll be able to understand which are the relevant variant that is causing this prediction. So the explainable part, which most black box machine learning tools suffer from, and I'd like to understand, how has this been useful for your team? Has it been accurate enough? Does these results make sense? The explainable part of the ML tool, was it helpful?
Yeah, no, that's a really good question. Yeah, so it's a good point to bring up. So that's one of the main reasons we like the random forest approach, and why we've incorporated random forest compared to sort of deep learning or other approaches that are very popular in machine learning, is that it isn't a black box, in that it's quite easy to look at the underlying forests and the trees built within the random forests as to which variants are interacting with each other. So rather than just saying, okay, this variant is the most associated with the outcome or the disease, we can actually see why and which variants, where within the trees in the random forest that these variants lie, and what they're co-occurring with. So which are the variants they're co-occurring with in the trees. And to answer the other part of your question, yes, so what we've done, so I haven't presented it today, but we're looking at a number of different data sets. So we're looking at a cardiovascular disease data set. We're looking at an Alzheimer's disease data set with VariantSpark. And specifically in the Alzheimer's disease data set, what we found is that the variants that are coming up with VariantSpark we're able to validate known Alzheimer's disease variants, such as the APOE variant. So that's a very commonly known variant. And we can see with our VariantSpark analysis that these top hits are coming up. But what we're finding additionally is other, obviously additional variants that are driving the disease outcome. So some we're validated from existing GWAS studies. So there's the GWAS catalog, where they've got all, for a particular phenotype, they've published all known GWAS hits. So we validated sort of well-known hits from there, but we found additional hits, and we're currently in the process of validating that. And as I answered the earlier question, really to validate that, we need to perform the results in an independent data set with a completely independent set of individuals who have the same disease phenotype. And we're finding that we do validate our findings. So that's work we're currently doing and currently writing up. So watch the space.
Yeah, that's wonderful. Yeah, that's wonderful. Okay, so I have another question from, I think this is Salvani. She says, thank you for the wonderful talk, Dr. Natalie. Does TRIBE use comparative data, for example, 1,000 genomes data or HapMap data to help the analysis or create the clusters?
I'm not sure.
I think she's asking.
Understand the question.
I think she's asking what sort of data is used as input for the TRIBE.
Oh yeah, sure. Good question. Yeah. Okay. So TRIBES is, again, it's a genomics-based tool. So all of our technology we developed is for genomic data. So the input, again, is whole genome sequence data or array-based data. So genetic variant data is used. The presentation, the examples I showed in my presentation in the ALS data set is from whole genomes. You can easily run the tool on array data as well, or whole exome data. But the input data is genetic variant data. So VCF files from genetic variant data.
Okay, on a related
1,000 genomes, found 1,000 genomes could be used. You could download the data from 1,000 genomes, run it on TRIBES or Wellcome Trust or HapMap. HapMap's array-based data, so you could use that as well.
Okay, nice. So just to clarify, do you need to have the sort of like parent-child
I've lost you again.
into TRIBE? Because I noted that you. Can you hear me now?
I can hear you again. You might have to repeat your question.
Okay. Just wondering whether you need parent-child information encoded when you're running TRIBE. Do you need to annotate the genomes? You don't. Okay.
No, so when you run TRIBES, the idea is that there's no relationship status known between individuals, and purely using the similarity between the genomes. So we identify these segments within the DNA, within the genetic data that individuals show that they're matching on. So they have high levels of similarity across chunks of the DNA. And by identifying, say, an individual who's a first degree relative, so a parent offspring or a full sibling, you can see that they in fact will share, quite a high degree of their DNA with each other, just by virtue of the fact that they are one meiosis away from each other. And this is quite apparent with the genomic analysis. You can easily see these regions of the genome that the individuals share with one another.
Okay, thank you. There have been a couple of questions from Mohammad Shanaji. Do we need manual screening of the results generated by these machine learning approach? As we deal with huge data, how can it be faster?
Okay, do we need manual screening? So I guess yes, because, it depends what you're using the results for. I mean, at this stage, these are still research-based tools. Obviously, we're also working with health providers within Australia, so health departments within Australia to use this type of technology for, say, predicting genetic variants of interest that a clinician might then use in an actionable mutation. So a clinician might then use this information for deciding how to treat a patient, for example, what drugs to use for the patient in a particular context, or how to further triage them in a clinical scenario. So we are working with health departments on this. So the technology would have to go through a series of steps to be used in the clinic, and obviously there's a lot more stringent. It's a lot more stringent when technology needs to be used in the clinic compared to be used in a research basis. So to answer your question, there is manual intervention in that you need to process the results that are output by the tool. But like with any bioinformatics methodology, there's a probability associated with every outcome. So with every genetic variant that's identified by the tool, there's a probability associated whether or not that variant is in fact associated with the disease or not. And so there can be a higher or a lower probability that variant is key in driving the disease. And I think the second part of the question was around how can we make the tools faster, right, with big data?
Yes, that's right.
Good question.
So we have a lot of data.
Yeah.
Yeah.
That's right. We do have a lot of data. And again, that's one of the sort of focus areas of our group, is to build technology with the future in mind, because as we know, the data is expanding, exploding, it's getting bigger by the day, the technology is getting cheaper and just, easier for people to get genome sequence data, and for this genome sequence data to increase in size. So that's why we've really taken advantage of the APBioSpark technology, because APBioSpark means that you can analyze, trillions of data points in a single analysis. Whereas if you try to use a standard HPC compute system or a desktop computer for this, you're really going to fail. And even some of the technology I've used, such as in GATK from the Broad Institute, or standard tools like VCF tools, I've tried to use these when you were looking at 40 odd million variants at once, and the technologists can't really handle this scale of data. So we really need a whole new generation of tools that is designed to look at millions and trillions of variants at once. So I think using the Apache technology, so the Spark technology, as well as cloud technology, which means you're not paying for the compute when it's not necessary, when it's not being used, is really the way forward for dealing with genomic data in the future.
Okay, thank you, Dr. Natalie. So I think we'll take 3 more questions before we resume for our group photo session. I believe we have a group photo session after this. So please stay online before we adjourn. There is one question on the chat box. If do you have anyone out there who wants to voice out a question? Please unmute yourself.
Hi, Hari. Can you hear me?
Hey, hi, Asif. Yes, Asif, go ahead.
Okay. I've been listening under a tree in the middle, in a rainy weather here. So that's how interested I am. Thank you, Natalie. I couldn't move from my spot
thanks for
because
dialing in.
the Wi-Fi was the best at this spot. Okay, anyway, thank you for sharing your corona analysis, and you showed that you identified 6 strains or something that was representative of Australian variants out there. I was just wondering whether you have done something similar for all the sequences, the worldwide sequences, and whether you guys have identified the representative strains for the world, and how much do they differ from the vaccine strains that have been used by, say, Moderna, Pfizer, and the others?
It's a very good question. So the work we published as I mentioned, so we looked at we did this sort of looked at the evolutionary landscape of the different viral strains present in Australia to identify sort of the key strains that we recommended for vaccine development. We did that work in around April, May, so it was pretty early in the pandemic. So there were a lot fewer strains present at the time. And so at the time, we we purely focused on the Australian, the virus is present in Australia. We haven't explicitly done this analysis for worldwide strains, but as this web service that we've developed, I mentioned, called a COVID Beacon, we've expanded to other countries. So COVID Beacon was a joint project with India, so they're looking at all of the strains available. All of the strains are present in the Indian population based from different centers around the country, which all belong to the Beacon Consortium. So it's possible to look at strains that are present around the country and variants that are present around the country. But we don't, we also, like I said, we also partnered with GizAid, so it's possible to use our web service to look at to run the analysis yourself. So to run the analysis to look at what strains are present around the world and what are the most sort of representative strains based on this 3D analysis. We haven't explicitly performed that analysis ourselves as such, because as I said, we've done this work back in April, which is when we published the paper, which is at the beginning of the pandemic. But it is possible for an individual to go and use the web service to run this analysis themselves. Then to answer the second part of your question, in regards to the Pfizer vaccine and Moderna vaccine. So again, because this is a more recent development, we haven't performed this analysis to compare the virus strains from the evolutionary analysis with what is being used in the Moderna and Pfizer vaccine. But it's actually a very interesting question, and I think it would be a really interesting question to ask of the data.
Thank you, Natalie.
Sure.
Okay, thank you, Asif. We have another question from Helia. She says, very inspiring talk, Dr. Natalie. For someone who has no access to university server or the university hasn't developed any server for genomic analysis, where do you recommend to start? She complains that she is using her PC and it takes up really huge space.
Yeah, that's a good, I think this is a common problem for many people across the world, and certainly certainly in countries or institutions where they don't necessarily have access to funding for maintaining a large infrastructure for dealing with this type of data, or smaller groups which are not, they focus on genomics, but the larger organization isn't necessarily dealing with large data sets themselves. So I think this is another reason why we wanted to develop these tools to be cloud-based, because individuals can then use the tools without having to have their own compute infrastructure. So all you need where I would recommend starting is really to log into AWS, so open an AWS account, and you can request the compute infrastructure that you need for any purposes, so any analysis that you want to perform. And rather than having to have the whole infrastructure available to you at the university, you can just pay for the resources that you need at the time through AWS. And I think they do have specific accounts for academic users, which are quite highly subsidized compared to commercial users. So I would get in touch with AWS or any of the other cloud providers for that matter, and ask them what they can offer you as an academic researcher. And in fact, work we've done with them, because we collaborated with them, we actually negotiated some quite competitive rates, and because they were interested in us showcasing that the work we developed is utilizing their technology. So there's always opportunities to work with these providers. I would definitely recommend that you use sort of cloud computing approaches if you don't have your own infrastructure at the university.
Thank you, Dr. Natalie. And I think just to add, if you have a proper proposal of your research, you can, if I remember correctly, you can apply for an AWS grant. I think they award you
That's
up
right,
to $50,000, if I'm not wrong. Yeah.
Yeah, exactly. Yeah. That's right.
Okay. Anyone else with questions? Please unmute yourself. We have a last question. Anyone? Going once, going twice. Okay, the session is closed. So please do not log off yet. I'll pass the stage to our MC.
Thank you very much, Dr. Natalie, for the wonderful talk. And thank you, Dr. Hari, for chairing the session. I will now invite everyone for a photo. Ms. Helia, please take the lead over the photo session.
Okay, everyone. Please help turn your camera on for the photo session. We will wait for a few seconds for everyone to turn on their camera. All right. Get ready. Smile. 1, 2, hang on, 3. Okay. There are several room here. 1, 2, 3. Okay. Hang on. 1, 2, 3. One more time. 1, 2, 3. All right. I think I will take one more time for the other group. Oops. Hang on. 1, 2, 3. Okay. Thank you so much, everyone.
May I say a few, make a few comments?
Yeah, Dr. Asif, I invite you to make few comments. Oh, the
No, that
Prof
was
Tan,
Prostan. No,
please go
yes,
ahead.
Prostan.
Go
Thank
ahead,
you
Prof.
so
Tan.
much. Thank
Yeah,
you
okay.
so much. This afternoon, I'll be having a meeting with the Chief Executive of the PAWSI Supercomputing Center, also from CSIRO, and with Sean Smith, Professor Sean Smith from the National Cancer National Computation Infrastructure at ANU, the Australian National Universities, together with the chief executives of many supercomputing centers. And we are trying to work out a scheme whereby ASEAN's participants can apply for access to high performance computing facilities in our respective organizations. So do look out for the announcements if we manage to agree this afternoon to provide computational access of supercomputers, high performance clusters, and possibly even cloud offerings to the academic institutions throughout Asia. May I also note that there's also international competitions. Some of you who are interested might be interested in looking at the HPC AI Advisory Council, HPC AI Advisory Council, which regularly organizes international competitions. And recently, as part of the effort to support COVID research, they have a component of the competition that includes solving structural problems related to COVID spike proteins, etc. So this is our effort here to promote high performance computing amongst the bioinformatics life science communities, in addition to other communities in other disciplines. But for this community in bioinformatics, we are working hard to build the infrastructure in order to support your research and to make it go faster and bigger and more effective for a transformational bioinformatics led by young people like all of you. Thank you.
Thank you very much, Prof. Tan. Dr. Asif, would you like to give some comment?
All right, I'm getting drenched, but yes, thank you, Natalie, for helping us inaugurate, being the inaugural speaker for our APBioNET talks, and thank you, Prostan, for helping us launch this, and to Helia for organizing and managing this. I hope the participants enjoyed the talk, and I hope they will look forward to more exciting talks from APBioNET talks initiative. And don't forget to subscribe our YouTube channel so you can get notified whenever we post some of these talks or more of these talks in the future. Thank you once again. Thank you for your support, and this was great. Thank you, Helia, and thank you, everyone.
Thank you, Dr. Asif. Thank you, Prof. Tan. Thank you very much, Dr. Natalie, for the enlightening talk, and Dr. Hari for helping with sharing the talk, and Prof. Tan, also Dr. Asif. Thank you, everyone, for attending today. We invite you to vote for the polling to help us figure out your preferences for next talks. Thank you very much for attending our first talk. Apologies that it went ahead earlier from our schedule. But you can watch the whole recording in APBioNet channel posted in Zoom chat. Chong will help post the YouTube channel for the recording. And thank you very much once again. And we hope to see you again in our next APBioNet talk. I hand it to Debangana. Thank you.
Thank you everyone for active participation. We have come to the end of our agenda for today. Thank you once again to everyone for joining in our first talk of APBioNet. We wish to see you again for the next talks. You can register on the link which will be given in the chat box. It was a pleasure to have you all. Bye and have a great day.